Welcome everyone to the MalariaGEN Journal Club. My name's Chris Clarkson. I lead the MalariaGEN team at the Liverpool School of Tropical Medicine. And today we're very excited to have Harun here, Harun Njoroge. He's an early career researcher with an extensive background spanning biotechnology, molecular biology of parasites and vectors, and quantitative skills in global health. And he then went on to do a PhD in vector biology and genetics at STM. His research focuses on uncovering the adaptive genetic mechanisms that enable insects to respond to environmental pressures, including pesticide exposure, which is an area critical to vector control and disease prevention. Harun's going to talk about the role of genetic surveillance in malaria elimination strategic plans in Africa. And we will get back together after talk, and we will be able to ask questions to Harun then. So yeah, I'll hand over to Harun.
All right. Good afternoon, everyone. My name is, as been mentioned, is Harun, and I've got quite lots of interest in studying how vectors adapt to changes. This was quite a good opportunity when I got my PhD. I had this opportunity to study at a countrywide scale, I would say, and that's what I'll be presenting to you today. What I would just maybe try to say is this paper is not really that I was looking for insecticide-resistant markers, but it was more of looking at a general landscape of how a whole genome would be able to adapt when exposed to two contrasting vector control intervention, and how at population level also the mosquitoes responded rather than just a single mosquito level. So I'll move on to the next slides. So basically, this slide talks about the key reasoning I have behind this work, and I think at the moment, not just in malaria control, but also other vector control intervention that rely on insecticide to control the disease, is we had a evolutionary landscape. The pathogens and also the vector themselves are evolving to the tools that we are using. And especially if I go back to the vectors, because that was the focus of my study, is LLIN, which is the backbone of vector control, and all these other new control tools, they are still reliant on insecticides. And as we continue using them, vectors are going to continue adapting to these changes. And so does us keep improving or increasing the amount of chemistry going to solve the problem of insecticide resistance? I think for me, it is more of working with what we've got and trying to think how to reasonably use the current tools that we have, and also new ones are coming, and try to extend their use or sustainability within the control field. And so what I wanted to address with this study is understand the impact of having combinational tools, or tools that have more than one active ingredient or an additive ingredient on the vector population, compared to the standard tool that we have. And in this case, I was comparing bedness, which is the main tool that is currently being used. So using standard pyrethroid bedness compared to the newer generations that are coming, how the population respond, and is it a solution to addressing the insecticide resistance problem. And what I need probably is good to note is I think if we're gonna proceed to eliminating malaria in most regions, it should not be just evaluating an intervention if it works, but it will be also important for us to understand how do they adapt to these tools, because if we deploy them and we don't understand how the vectors adapt to them, we'll end up having a scenario where we are saying malaria indices are not going down, but we don't really have an explanation, and we now have to do forensics to go back and try to understand what really happened. And I think being proactive is much better than being reactive. And so what are the best ways to evaluate, I would say, the tools that we are deploying in the field, be it in a trial or be it in a control setting. And I think what I've highlighted here, the main evaluation method that we have and most programs really care about is, are we reducing the parasite prevalence? Is malaria going down? Are we seeing less anemia in kids? Because that's basically what we want to see, that we are getting rid of the disease. But there are these other measures that are equally important that also are addressed, and not always addressed, but sometimes they are addressed within an intervention, but in trials they probably do address them sometimes, and these are the vector indices. And we look at in terms of entomology, we look at the mosquitoes density going down. Is the mosquito density going down? Is the transmission based on entomological infection rates going down? Or in terms of species, are we killing one, replacing the other? And so all these things can be able to tell us different, and there are more of like different layers of information that can be able to help us evaluate how the tools is working. And in addition to that, at least at the moment, some of the control programs are looking into product durability, because it's important. If, let's say, the vectors are not going down, is it the fact that the product that we've put in the market is not durable, it's not up to the standard? So we look at that as well. And an important key also is when we also evaluating this intervention or trials, we need to also to look at how the vectors actually themselves respond before the intervention and after the intervention to these tools. And by that we use bioassays, which of course we able to sample the population in the field. And I'll talk more about that in terms of how we sample and if it's really representative of the population. But when you get a representative and we're able to see, are they really dying or not, that also tells us how the vectors are responding. And it's also another layer of information when it comes to evaluating trials. But the problem with bioassay, which as much as it's quite nice, we do have a problem. And the biggest problem is how variable it is. And for that, I will be able to show some data on some series of bioassays I did when I was doing for another study. And this, as you can see, the batch is a different bioassay day that I was doing and a different population that I collected. All these mosquito passes were collected in the same village within 5-kilometer radius. But every other time I did a bioassay with 2 different concentrations, this is based on CDC bottle, you can see how variable the outcome was. So the red means they are dying, the mosquito that died 24 hours later. The blue is whatever that had been knocked down after 1 hour exposure but recovered later. And the green is where the mosquitoes that died completely, that were alive, sorry. And so if you look at the lower dose or the higher dose, you can see how variable bioassay can be. And so, for example, if one day you were able to see most mosquitoes surviving and you use that data to inform policy, you'd say the mosquitoes are resistant. And then in another day, you find high level of mortality and you say the mosquitoes are susceptible. And so this is one of the major limitation of bioassays. And also another limitation of bioassay is, how do we sample the mosquitoes that we use to represent this data? Do we just sample in one house or in one paddle, and we assume that represent the whole population? When you go and collect the mosquitoes, go to the lab and do the bioassay, would find, well, there's less than 90% mortality, and so we say the population is resistant. So by luck, if we go to in one paddle and we find all mosquitoes are dead, we say the mosquitoes in that population are susceptible. Is that the right conclusion we can make if we don't have a strategic or data-informed or sort of like a well-thought surveillance method that we can be able to collect in different regions in that area and do the bioassay so that I can say this is representative of the population? That can be a challenge because bioassays are quite intensive. And that is where I'm going to go next. That's what people think about, using molecular markers to be able to look at insecticide resistance. And in 5, the population itself is resistant or not, because with the marker, you can be able to collect mosquitoes, archive them in the lab, and then be able to do just one large genotyping procedure and be able to tell the frequency of the marker and then relate that with the resistance level in the area. But again, the non-resistant markers can only be able to tell one story. Right now, we are using so many interventions, so it's not just pyrethroids. We are using different interventions. Will they really give us the information that we need? And this is where I see that there is a surveillance gap that need to be addressed. And this is where, in terms of genomics, we need to think about how genomics can be able to address this gap. Because yes, non-markers can be able to tell us if, let's say, KDR can tell us if the mosquito are susceptible or resistant to pyrethroid to some extent based on associations, and sometimes the association is not that clean. But beyond that, at the whole genome level, what is happening? And I'll give an example with a paper we did, and or sort of like a study I did in East Africa, where I was looking at the population. We know in East Africa, the population has a huge portion of the resistance is contributed by metabolic resistance. And as much as we do a lot of KGR monitoring, it doesn't give us a whole picture. And when I looked at the history of a triple haplotype or mutant that is linked to the RP1 or that huge loss that is associated with pyrethroid resistance in East Africa, was able to find there were some 3 linked mutations that were spreading in the population very fast. They started as a 2-linked mutation, and then they become 3 when you have a duplication, and then it spreads so fast within East and Central Africa. Within 5 years, it had reached all the way to DRC from East Africa. And if we didn't look at that point with genomic surveillance, that picture would have been lost completely. You would never have known about that. And so what I think is that the trials, most of these standard endpoints that we always evaluate, will always tell us if malaria has been reduced transmission or if the density of vectors is going down, but they don't really tell us if the population structure is changing and what the intervention is really selecting in the population. And that was a really key of this is a paper I was talking about for those who are interested to read about. And that was what really drove or the main basis of the study that I did, and the paper that I'm presenting today is if genomic surveillance can complement trial or vector control endpoints by revealing how population will be adapting to all this, the intervention in place, and using that to connect with the other the other measures and be able to make sort of like an informed choice or an informed decision when we're moving forward in control methods. So my study was based in Uganda, and I was quite lucky to have access to samples from the lineup trial, which was a very large national surveillance or distribution campaign where the lineup trial was based. And so with this kind of a trial, it allows you to have access to a huge population. In this case, I think I would say so nearly a third of Uganda. And being as a trial, most of these clusters or districts were all randomly assigned to the 4 types of badness that were being evaluated. It's like 2 main badness, either PBO or non-PBO, but also there were 2 different companies, Olicet or Permanent. And so I had entomological collection that were collected at baseline before the trial, and 6 months every other 6 months until 25 months later. And so with this kind of data, it sort of like allowed me to have a huge power to be able to look at evolutionary process in the field and at this large scale that probably previously people have not been able to look at. And with this kind of trial, we would have been able to answer this question that I'm asking: can genomic surveillance really add anything beyond what the standard measures did add during the lineup trial that was reported by the authors of the lineup work? And so lineup study itself had already established very important findings. First of all, was an entomological impact. They were able to find an epi where non-PBO nets were, I would say, a bit inferior to PBO. So PBO was able to show much more reduction in prevalence of malaria, parasite prevalence, and also a larger reduction of densities, vector densities compared to the standard badness. And also another follow-up study by Amy and other authors, they were able to look at common known markers, and they were able to demonstrate some of these markers like the VGC, the KDR or VGC markers were increasing in frequency, including the triple mutant haplotype markers that had already found in that population around there. They're all increasing. But the biggest challenge is with that pre-genomic study could not be able to differentiate what was which intervention really, the contrasting difference between the two interventions. So they're all increasing in frequency, but what did each intervention do? And I think that was also what was limited in this studies. So that's why I ended up doing this genomic study, to be able to see what could have been missed out. So this was the study design. I didn't collect the samples, neither did I do much of the work of sequencing. So much of this data, much of this work had been done by the previous studies. What I did was to select samples that had been sent for, they've been sequenced and they were in MalariaGEN platform. Look at the samples at baseline. I looked at 6 months later, but the samples were quite few, so I could not be able to do very good contrast between the two time points. And all through the other time points, I could not be able to get decent samples. So I sort of like ended up deciding just to compare the baseline and the final collection, which was 25 months later. Does it make a difference? It could. Maybe there are some things that we miss out in between, but also some of the signals, population genomic signals, they take a while for them to become much more clear. So a few generations will have to change for us to see some of these changes. You'll not probably be able to see them immediately. But probably could have been much better if we had a good time series and be able to look at these other regions. But that is one of the biggest limitation of this study, that I didn't have enough samples in between. And it was more based on how the lineup trial was designed. It wasn't really designed to be more of like a genetic study, but it was designed to meet the epi and entomological indices. But in terms of genetics, we could have need to do, when we have months that we have low collection, we could have had to increase collection effort so that we have at least enough samples, representative samples for each time point. And so there are two key main things I wanted to look in this study. First of all, was to look at the demography and diversity. So was there really a huge change in terms of I would say the breeding potential of the population, which most of the time people call it effective population size, which is effective population size and census population. They're quite different. Census is just, if you were to count the mosquito one by one, that would be the census population. Effective population size is most of the time smaller, because this is a measure of which the number of mosquito in the population that are contributing to the next generation. And most of the time, they are fewer, because not every other mosquito would contribute to the next generation. That's the simplest way of explaining effective population size. And in terms of diversity, it's just a measure of how the variations we have in the population. And so by looking at these two measures, for example, if the population has really been crushed, we would expect to lose a lot of diversity in the population, and you'll be able to see that signature in the genome. And also in terms of effective population size, if, for example, the change that happened in terms of the densities reported in the lineup data, if it mirrored with effective population size, that we had killed so much that even if reduced the breeding potential of the population, we'll be able to see that signature in the population. The second base that I looked at is how the population adapted to these two types of bed nets. Did the current resistance markers of mechanism continue increasing probably in the standard bed net areas? And how did they respond when you introduce PBO bed nets, which also had the addition of PBO, which would be hitting down really hard the cytochrome P450 mechanisms that contribute largely resistance in East Africa. So how would those population respond to those new bed nets? And with that one, I looked at through a normal allelic frequency study, looking at the frequency of SNPs before and after. And then I also use FSTs, which is population differentiation, if I could see any differentiation between the two time points. And the most important one is to look at level of selection. So there are areas that are already selected. If you look at the frequency of those selected haplotypes before and after, you can be able to see if there was a change in some of those areas. And those were the key outputs that I expected, and some of the interpretation I'll put here, like to see what would have been. I'll talk more into that as I proceed with the presentation. So I'll start first with the population structure. So for any population genetic study, it's good to understand the population you're studying. So is the population homogeneous? By homogeneous, I mean there is no really huge difference between population from, let's say, Eastern Uganda and Western Uganda. They are quite similar in terms of population genetics at population genetic level. So if they are quite different, then it means you need to think about how well you're going to handle this population, so that difference is not going to be able, it's not going to sort of like take out or be the background noise that takes away the actual signal that you want to compare with the intervention. And I use two methods to look at the population structure. I use the principal component analysis. And as you can see, the top panel with PCAs, much of the separation or differentiation of the population or the differences were driven by inversions, but not really by the population being collected or the mosquito coming from Eastern Uganda or Western Uganda. And that, just based on PCA, we would quite say the population is largely homogeneous, only varying by the inversions, not really by location. But when you look at a more sensitive method to look at the population as well, like using a whole genome scan, FST scan, and it's a bit windowed, you can be able to see, yes, as much as the population is homogeneous, but for some of this region, especially the resistant associated sweeps, where you see these peaks is where you always have the resistant loci that are swept in the population. And you can see when you compare Eastern and Western Uganda, where you see the green, it means there's significant difference in terms of those genomic regions, and you can see there was significant difference between those two populations. So as much as there's extensive gene flow between the populations across Uganda, also there's this rapid changes that are occurring in resistance. This is interpretation that I have. I could be wrong, but in this resistant associated loci, there was rapid changes that made the population look different. And so for that case, it meant that for all subsequent analyses that I did, I had to account for location, despite the population being largely homogeneous. And so this is the first main results of the study. So here I've included the findings from the lineup trial, showing that when you compare the densities, the cot densities in-house from the houses indoor densities, there was a significant difference between the different time points. So at 6 months all the way to 25 months for both nests, you can see there was a reduced density or relative density in the collections in the trial. But it was quite large. The difference is quite large, as you can see with the PBO bedness. And so that is what lineup study concluded, that at entomological level, the density reduced. But when I looked at population genetic level, the diversity in terms, when I was looking at nucleotide diversity, and also I looked at, of course, sorry, I got distracted by the chart. And you look at the effective population size, I did not see any significant difference at baseline, and also comparing baseline and post intervention. And the problem also with LD, like for example, even I looked at the LD itself. So I don't know how much people understand about linkage disequilibrium, but it's more of like a random association between 2 points in the genome. And if the population is quite small, most of the individuals will be breeding with each other, and they will tend to be quite similar. And so there will be this random association just because you're breeding, like for example, a brother and a sister, they would have a high LDL between their chromosome just because they are related. But if they were to, because you're breeding with us and there's high rates of recombination in a big population, the LD becomes quite small. And so when you looked at that measure, and then I use a linkage disequilibrium to infer effective population size, both measures, I could not see any huge difference. And of course, one of the critique I got from the reviewers was, did my study have the power to detect that? And we did some simulations. And based on the simulation, we looked at a small population from around 10, if the population was around 10,000, 100 actually, all the way to over 100,000. How much power would we have to detect that kind of change at the sample size that we had? And we could be able to detect 10% or 10-fold change if we looked at a population that were less than 10,000. But when you start getting at large population above 100,000, even if we increased the samples considerably, we didn't have enough power to detect 10% fold change. And we know mosquitoes, they are in millions in terms of population size. And so even with a 10-fold difference, that we didn't have the power to detect that change. And so probably there could have been changes, but we couldn't be able to detect with the effective population measure. But again, I need to qualify this bit about indoor catches, because the way we sort of like evaluate using entomological measures, sometimes we don't really think about what we are really measuring. For example, the indoor collections, we have 2 nests that are working. And so what I always wonder, because we are not actually measuring the density of the mosquitoes that are going into the house, we are really measuring in the morning, how many mosquitoes are we collecting that are alive? And that is what we are aspirating. What about the mosquitoes that went to the bed net and died? We don't collect those ones. And so at the end of the day, we'll say we've collected a reduced number, but it's really probably not really a reduced number, because the number of mosquitoes that went into the house the previous night were probably the same, but some of them died because, of course, of the intervention. And because we are not accounting for that, we'll always say the density is going down. Probably the density is going down, but I feel because the population is being killed, and actual population is the actual number of mosquitoes we collect is just what is alive, that the dead ones will never be able to tell. And so this sometimes makes me want to think that, you know, that data, I don't know if people have been able to read the spatial repellent paper from East Africa, where they saw epi outcomes, like the malaria went down, but vector didn't go down, but they were measuring that with human landing. For example, if we were to use human landing in the lineup study, would we be able to see a reduction in density? Because with human landing, we are getting the actual mosquitoes that are coming to us as a human. And so we'll be able to compare that before and after. Will the numbers go down really? Or is it just something more of like a limitation with the indoor collection method that they use in lineup? And so that could also be an explanation. That's why we are not seeing really a huge difference at genetic level, in addition to some of the limitation I've talked about. I'll go now to the next key findings of this study, and this is where we are looking at adaptations. And with this one, I used 2 key methods. SNP level changes in frequency. And here I present a Manhattan plot, because I use more of like a GWAS kind of scan across it, because it's just a genome wide. So it's millions of SNPs that I'm looking at a point and comparing their frequency before and after. And I had 2 hypotheses. One was to look at the change, overall change, regardless of the bed net. Like, was there a change in frequency driven by the intervention, the bed net itself, despite if it's pyrethroid treated or not, pyrethroid treated, or it has an additional PBO. And with that, for that hypothesis, we were able to see there were around 21 SNPs that were changing in frequency. Most of them were intergenic, not within genes. It's only one that was in a gene, and it was synonymous. Others were intronic or non-intergenic. And I did a bit of functional validation by looking if they were SNPs found in regulatory regions. Maybe they were associated, or they were changing because they're driving the change in gene expression. And so I did that. And I was able to see some of them. Most of them, they were not associated with a regulatory region, apart from one that was found in a distal CRA, which is by this I'm just talking about those cis regulatory elements. So this is something maybe can be looked, but I didn't really go into details with that. The second hypothesis where I look at net specific changes, and at this I didn't find any SNP that was significant or was changing significantly in either pyrethroidness only, or the ones that had PBU. And I guess it's because the sample size was much smaller, and so I didn't have enough power to detect. And I did that. Of course, we tried to test that based on the smallest sample. This is a PBO cohort that had the smallest sample. We only had power to detect over 29% change in frequency, and that's where probably this explains why we didn't see anything with that when I tested that hypothesis. And so with this method, I could not be able really to tell if or resolve an specific evolutionary dynamic, but I was able to say the badness really had an impact on the whole population just based on the few SNPs that changed. But again, some of these SNPs were changing close to the centromere, so it's a bit, it was really difficult to interpret the results, and that's why I did a windowed analysis. This is the second results that I'm presenting here. And I focused on swept regions. And so I used statistics called H12, and it just looks at the frequency of the most common frequency, the frequency of the most common haplotype that are being swept in a population. And so what I did was to look at the frequency of those swept swept haplotypes before and after, and then get a difference. So that's why I have the delta H12. So I looked at the difference between the two. And so I don't know if it's very clear, but if you see a peak that is positive and it's got a green, it means that was significant. The SNPs that were changing, the haplotypes were changing significantly, and positively, it means they were increasing in frequency because it was after minus before, so it means they're increasing in frequency. And if it's facing the downside or the negative side, means that this those haplotypes were reducing in frequency. And so four key is sort of like results you need to get from here, and I'll just talk you through them. This is when I was looking at net specific change, and I looked at I've highlighted 3 regions that I saw they were net specific. A key one is a C9 cytochrome P9K1 found in the X chromosome. And I've presented here some of the graphs just to visualize that in terms of looking at the actual frequency of the P450 duplication, because I found that the haplotype, the swept haplotype, is strongly linked to the duplication of that gene. And when you look at the frequency of the duplication in the badness, in the standard badness or the pyrethroid only nets, the frequency was increasing before and after. You can see there was a slight increase. But when you look at the PBO arm, this is East Africa, you find there was a decrease in frequency. And then in Western Uganda, you look at that, the same on the standard badnet, the frequency was increasing in the standard cohorts, but when you look at the PBO cohorts, the frequency was going down. So it's like the frequency of the cytochrome P450 duplication, or the haplotype with the duplication, was going down. And this was quite good because it was more of like a replication of the results, independent populations showing the same trend. And so for me, I ended up concluding this is an S-specific change where we're seeing the frequency of duplication of this cytochrome P450 increasing where we have only pyrethroid badness, but where we have PBO in both populations, the frequency was going down. And of course, there's so many interpretations you can come with that. One of them that I argued was when you use PBO and you've knocked down the P450's ability to sort of like break down pyrethroids because you've inhibited them, then that haplotype stops being very important and is not going to be maintaining the population at the same frequency compared to where the same duplication is sort of like giving an advantage, because the P450 having increased copies means there's increased copies of P450 that are breaking pyrethroids, and so you tend to select for that haplotype in the population. The other loci was in a region within the 2LA inversion. So we call it 34MB because we're not very sure what is driving that sweep. And this one is not as clean as this one, where the only change we were able to see in the standard bedness, there was no really consistent change. So that one in both population, there was no significant change in both of them. But when you go to the PBO only cohort, in both East and Western Uganda, we find there was an increase in frequency in both population, and so somehow the haplotype was increasing or was being selected in the PBO cohorts. The DGK is not that it's not that strong evidence because it was only present in Western Uganda, but in East it was not really changing. So that one I won't go much into details. Another key result that I want to present here is in terms of region-specific change, and this is what I've highlighted with the VGSE region. And if you look at the VGSE region in Eastern Uganda, in both PBO and non-PBO cohorts, the frequency of the VGSE haplotype was increasing in frequency. And something I'll demonstrate, I'll talk about here is that in Eastern Uganda and in Kenya, the haplotype that is in high frequency is what we call the KDR East, or the S haplotypes, the S2 or 3, most of them, and they are quite in high frequency, and they were increasing in frequency in Eastern Uganda. But in Western Uganda, which borders also the DRC area where we have much of the F haplotype, it seemed that the frequency was going down of the main haplotype. And this is where I would say as you can look between the pie chart, the S haplotype or the Eastern KDR East was being replaced by haplotypes that were from the F background, the KDR F that we talk about. And so this is more of like a region-specific change, and we were thinking is that there was an inflow of the F background from, or it was still probably there in low frequency, as you can see, but because it's sort of like, it gives more, like F background, we say has a stronger effect in terms of resistance. It was being selected over the KDR East haplotype, and that's why we are seeing this contrasting bit. And because probably we don't have a high population of that, you can see even in Eastern Uganda, the F was starting being selected, but it was not in very high, so that's why it didn't really affect the main haplotype that was very high in frequency. But still, it was somehow selected even in the East if you look at the pie charts. In terms of the some other regions like the main RP1 region resistant loci that we always find all the major cytochrome P450s that contribute to resistance, like the P4, P3, we didn't see a clear net or region specific. So in both of those both regions, the frequency of those haplotypes were increasing, and this just sort of like mirrored the results that Emily presented or published in her paper with the known markers. The frequency increased in all loci regardless of the net type or region. And for GST, which I thought probably would have increased in PBO areas to just counteract the PBO effect, I didn't see any change in both cohorts in both regions in terms of a change in frequency of that selected haplotype that we find in the GST region. So the main interpretation here is that at least with haplotype methods, we're able to see net and region-specific signals that were really missed with SNP-based tests, both at genome-wide level and also with the markers that Emily used. And so this sort of like give the value of doing genome-wide surveillance in some of this work that we're doing. But I think the biggest problem here will be to discuss about the cost of doing this, and if there are other methods that we can be able to use that can be able to give us this kind of a picture and still be able to report a kind of have this kind of additional results when we're doing normal program intervention or trials. So what my study added beyond the lineup bit, I've probably gone through that during my presentation, but really what I showed is sometimes entomological metrics will not match population genetic metrics. And so this is an area that we will try probably to harmonize how we do our collections and see if the way we do our surveillance or monitor the entomological metrics can be able to match what we can see at population genetics level, or probably they will never match because they're measuring different things. In terms of intervention effectiveness, I've sort of like shown how we can be able to hijack big natural experiment, and these natural experiments are happening all the time in Africa. When you're doing mass bednet distribution, mass IRS, we just need to be able to think about how well you can be able to evaluate user platform to be able to collect data that can be able to tell us how the vectors are evolving. And this can be able to, it's more of like what I was saying, it's a proactive way of sort of like catching a problem before it becomes a problem, and or getting some explanation for what we're able to see down the road after we start seeing the intervention not really working well. And so I would say genome-wide surveillance is quite valuable when you embed it in intervention and also in programs. And this can be quite important or useful when you interpret it alongside all these other trial endpoints that we measure regularly. And also it can be able also to help us with, making operational decisions. And I'll go more into details about that in the next slide. So I've gone through a lot of, try to make the paper quite simple, not to go so deep into population genetics. And if I didn't manage to do that, I think the 3 take-home message everybody should go with is the interventions. The interventions can be quite a selective environment, and I've been able to demonstrate within just 2 years, we were able to see PBO and non-PBO shaping the mosquito genomes differently, and the population responded differently. And so this is quite a big natural evolution experiment, and we can be able to measure and be able to see this story, add this story to our normal story about how the intervention are working by just using just an additional measure of genomic surveillance. And also this is addition by saying that we can be able to reveal stories that are hidden that will never be found with other standard measures. And finally, I think genomics is a useful tool when it will come to surveillance, sort of like elimination, that like we're trying to eliminate, and how we're able to use our limited tools effectively. We need really to understand how our vectors are adapting and how we can be able to do rotations or sort of like make decisions on what we can be able to deploy in the field. It is my paper, so in every journal club, we critique a paper thoroughly and sometimes someone calls the authors all kind of names. And so, of course, I critique myself not as badly as we do in a normal journal club, but I'll just point out some of the limitation of my study. Huge one was on the sample size and a bit of the power to detect some of the changes that happened, especially in the PBO cohorts. Some of the population are quite small. We couldn't, like when you do allelic changes at SNP level, we didn't have enough power to detect that based on the sample sizes we had. And also it could have been nice if at 6 months post the intervention, which is around 6 generation after deploying the intervention, if you count every month as a generation, 12 generation at a year or 18 generation later, I think there could be some signals that we may have missed out by looking at 25 months later. And so maybe in future studies, we can be able to do increased collection or use a method that can be able to help us get enough samples or representative samples for all these time points. As much as this was based on a clinical trial, and they're quite good at trying to reduce as much biases as possible by doing randomization and making sure all regions are okay. Still, there could be some things that we cannot be able to control. For example, mosquitoes cannot, as much as they were in PBO standard area and non-PBO, you couldn't stop them from migrating. And so some of them may have intermigrated between the cohorts. And so that could have diluted some of the signals that maybe occurred, and we could not be able to account for that. At each cohort, as much the areas are close to each other, some of them may be agricultural areas, others are non-agricultural areas, they're urban, and so they would have differences in terms of how the vectors would have responded to those interventions. Although I did more of like an average, like averaging all PBO cohorts and all non-PBO cohorts, but probably when you looked at smaller clusters, there could be huge variation in terms of changes in these results that I'm presenting today. Of course, the temporal resolution, that's why I was saying it's like I have a few time points to be able to compare and look at how this change, the speed of how the change occurred and how if there was persistence in between, or, whatever I saw at the very end, was it the same story throughout the different time points? Of course, the other bits are more of like a marker discovery and phenotype link, not really necessary at that level, but still it's more of like quite good to understand, especially like the 34 MB. What is really, is it just an association that we are seeing, or there's really a biological link to the results that I see? For the 9K1 region, because it's a P450, it's almost clear to say that there is a close biological link, but there are some areas that we don't really know what was really happening, or it was just a random change in terms driven by drift or something like that. And so just to finalize my talk, I think this is something people have always asked me, do we really need to do genomic surveillance, and what is really important to it? Yes, we will know the mosquitoes are changing, but how does that really change, or what is the value of that information to surveillance program? If the surveillance program are able to tell if the mosquito are dying or not just using bioassay, do we really need to use a very expensive tool to understand that? And I think I've sort of like tried come up with 4 points to say how well we can be able to use surveillance and make it actionable in terms of program use. And so, for example, based on my study, when we see a rapid rise of haplotypes or CMV immediately after deployment, that at program level, I think it could be a way to trigger intensified phenotyping and sort of like to be able to understand in those areas, for example, the PBO areas, we are seeing a rise in this frequency, we can be able to do phenotyping and be able to link the haplotypes to the phenotypes of how mosquitoes are responding. And they really do that change. Does it really change the ability of the mosquitoes to respond or to die because of this intervention? Another bit is in terms of product intervention. For example, if we've been using one class of insecticide, let's say the way we use chlorothiazide for IRS, and after a while, most programs were saying they're not really seeing an impact, and then they brought the actelic. When we have this kind of two different changes, can we be able to look at genomic level to see how, is it the mosquitoes that are responding, or is it just something to do with the product that is not quite effective in terms of its remaining stable in the population, and probably the mosquitoes are still susceptible to both of them. It's just that it's something to do with the product. So that can also be able to inform in terms of procurement rotation, and also how the population responds. If it responds quite rapid to, let's say, a tool, and we look at their actual genomic regions associated with that response, then it will say like, well, it's time for us to rotate with another product, because we can see actually the mosquitoes are really responding. It's not just at phenotype level. And for example, so for some of these novel regions that we don't have any in our panel, in our markers, we don't really know them. And if every time we do genomic surveillance, we see they are repeatedly changing in terms of frequency, this would prioritize them for validations. And that is, I think, to me, is important, so that we can be able to proactively be able to add new markers to our panels of doing molecular surveillance, which is much cheaper than whole genome sequencing. And basically, a program is more of like learning, being able to integrate all these signals with the information, can be able to sort of like guide us to understand the whole outcome of an intervention. And I guess with this, if we set this kind of threshold, surveillance, whole genome surveillance become important at program level. So that is the end of my presentation. I've tried to rush a bit, give some time for discussion, but I didn't keep much time. But I really tried to explain as much as I could to make it clear. And I would like to acknowledge all these people that were important in this study, most importantly, the lineup collaborators and the source of the funding and LSTM. Thank you.
Thanks, Harun. That's super cool. It's really awesome to see the genomics of the vector going alongside a trial like that. If folk have questions, I think you can either put your hand up or you can write stuff in the chat, and I can pass that on to Harun. I have a question. It's not a very nice question.
Yeah.
So you pointed out earlier on that there's this issue. So there's an ongoing trial. You expect the mosquito population to collapse. So there's that bit
Yeah.
while the trial, you know, when these new nets or new control method has been introduced, and
Yeah.
you know, you're just not able to collect enough mosquitoes, live blood-fed mosquitoes inside houses.
Yeah.
And so, you know, do you increase the effort from a constant effort to some sort of increased effort? But even then, you're collecting the mosquitoes. You know, your hypothesis is that actually you're only collecting the ones that survived. A bunch of them came in the house and died, and you're not measuring those ones, right? So
Yeah.
then you've
Yeah.
got an issue of detecting the sort of population allele frequencies if you're only
Mhm.
collecting the live ones. Is there anything that you've thought of? I just sort of the experimental huts came to mind where there are ways of sort of measuring mosquitoes coming in and leaving. I'm wondering if there are any other ways of collecting samples for genomics that you could start to get around some of these issues.
I think when I was discussing this a while back with John, I was thinking like if we could be able to target where the mosquitoes are breeding, it could be a quite good representative because at that point we are not really doing selective collection. Like there are some mosquitoes that will not come into the house. There are some that enter and just repelled immediately, they disappear. Some of them are dead, you'll never account for them. But if you could be able to go to the larvae and collect the larvae, do a very good representative of different areas because of course a larval habitat also increases another problem of collecting quite closely related mosquitoes within the same pool. But of course, if we account for the seeds within that and do a quite good surveillance, I would say that is a better method to use for genetic studies because you'll be almost collecting, I would say, a representative of what we have in the population. So I think other than that, unless if we use different methods, we say there's going to be always a bias. If we use, let's say, human landing, it's going to be only the mosquito that are attracted to the human, not all the others that are not attracted. If we use CDC light traps, outdoor and indoor, again, there's going to be that bias of with the CDC light traps. I feel for most genomic studies, if it's possible, I would say larva would be a quite good source of a representative sample.
Oh, thanks. Do we have any other questions or thoughts? We did have a request from Ditchell. If you could share your presentation, share your beautiful presentation. It is a beautiful presentation. I think, yeah, reach out to Harun maybe after the talk. But also, I just wanted to say that we do put up a recording of the journal clubs on our YouTube channel, so you can also find that later if you want to come back to it. There's a question from Richard. A really nice example of how you can understand selection in a natural experiment. The finding that CYP9K1 duplication frequency increased when standard bed nets were used, but decreased with PBO as core. Were these changes statistically significant? And did you need to correct for multiple testing?
Yeah, so they were significant because we did it at haplotype level. So when I looked at the frequency comparing, it was significant. And because it was just a single loci, I didn't need to do multiple correction because I just focused at the actual haplotype or the duplication, the frequency of the duplication. But in terms of SNPs, where I was doing thousands of them, we did just the multiple correction as we do in GWAS, and that's why it's only 21 SNPs that survives that brutal tests.
Just what's your hypothesis for the decrease where seen in with the PBO nets, the cytokine 3450?
I think for me, it was more of like what is important will be we remain or we increase in frequency, and in terms of PBOs being inhibitors. So if you inhibiting the P450s, they don't give any advantage to the population anymore, and so that swept haplotype will start disappearing in the population over time. But if the P450s are giving advantage by having extra copies in terms of like the 9K1, then it means that haplotype will continue increasing in the frequency, especially in the non-standard, the standard bedness. So I think that was a hypothesis I had for that loci. For this other additional loci, it was for KDR, it was more of like F being stronger than the West one, and that's why we're seeing an increase in frequency. But for some of them, it was difficult. For example, the 34 MB sweep, we don't understand what is being swept. And so I don't understand why is it increasing in PBO only and no change at all in the standard bedness.
Interesting. I wonder whether it's something like RNA-seq or something that could be used to dig into maybe what's going on there if we're not detecting it with WGS.
Yeah, it's also a bit complicated because it's within the 2LA, so it's quite tightly linked to the inversion. So I don't know if it's an inversion thing or what it could be. It's quite difficult to tease out actually that sweep from the inversion because they're tightly linked with the standard. Actually, it's increasing in the standard form, not in the inverted form. Yeah.
Interesting. Cool. Well, we're almost at time. Is there any other questions? Well, thanks so much, Harun. That was really cool.
Yeah.
And thanks to everyone that came and listened.
All right. So
I don't know if I'm. Thanks, Harun. I don't know if I'm advertising the next journal club, but I'm sure that there will be an email going around shortly with information about that. Cool. Thanks, everyone.
Cool. Thank you. The slides are with Kesh. She can share them.